Bismillahirrahmanirrahim.
Students, Assalamu alaikum warahmatullahi wabarakatuh. Welcome back to AmreenaMehrajTutorials. Dear students, today I'm again in front of you with yet another very interesting and important topic as far as class 12th syllabus is concerned. I'll not keep my lecture confined to class 12th. It will be useful for NEET students, those who are preparing for different medical entrance examinations, and even for students who are in higher studies. I'll try to make this video useful for all sort of students, be it the foundationary class 12th students or the students who are pursuing their higher studies. This will be useful for all the categories of the students. Now, my dear students, before starting this video, I would request all of you people to just smash that subscribe button and give this video a like, a heart and a share if you find it worthy, okay? That's very, very important because I just want this AmreenaMehrajTutorial family to grow, and to reach out to more and more students. That's possible only because of you people who are watching these videos, because I will not be able to, circulate these videos so much. It's you people who can share these videos and make others, capable of learning if you find it worthy, basically, okay? Now, my dear students, as far as the topic is concerned, it's again going to be the Mendelian single gene disorder. I've already uploaded one video pertaining to hemophilia. That was also a Mendelian single gene disorder because the mutation happens here in terms of just one gene. Only one gene is mutated. Okay, that's why we call it a Mendelian single gene disorder, because it follows Mendel's laws of inheritance as well. Now, my dear students, we have different categories of Mendelian single gene disorders. We have the X-linked disorders, we have the autosomal disorders, we have the dominant disorders, we have the recessive type of disorders. For that, you need to go and watch out my previous lectures. Right now, this video entirely is going to be associated with the X-linked disorder. Okay, once we use the word X-linked disorder, we get an idea that the mutation is happening or has happened or will happen to the X chromosome, which is the sex chromosome. That's why I use the word X-linked disorder or the sex-linked disorder. Now, once we use the words recessive, we need to understand it further, because I've already made you understand. Since we are going to take up another recessive disorder, you must have this idea clear. Recessive is such a type of allele or such a type of mutation which will be expressed in only one condition, when there is a homozygous recessive condition, okay? Under those situations, recessive allele gets an opportunity to express because we do not have a dominant allele to mask its expression, okay? So, recessive alleles express under homozygous condition. But who suffers more is the male, because if male has some recessive mutation on his X chromosome, there's no counterpart, there's no dominant allele going to be available for masking up that mutated allele. So if male suffers from some X-linked mutation, that recessive mutation gets expressed automatically. This is what we have understood in hemophilia, and that stands well for this particular disorder also. If you haven't understood this, if you have some confusion with this particular understanding, you'll be able to understand with me, inshallah, today. So, my dear students, the topic for today is color blindness, and you must be able to look at this structure of the eye, which is drawn on the board. I didn't want to waste the time drawing this picture for around about 4 or 5 minutes, so I've kept it ready. Now, since the topic is color blindness, so which is the particular organ of the body of a vertebrate, specifically the human, which is going to basically perceive or recognize the colors? It's the eye. So in this particular disorder, the defect will there be in the eye. The first important thing, okay? Now, my dear students, what sort of defect is it as far as the name indicates? The name of the disorder indicates that in this particular disorder, a person is not able to distinguish between the colors. So I'll write down, in this disorder, the person suffers. So in this particular disorder, an individual, I'll write down, an individual is unable to distinguish between, specifically I'm talking about between red and green color, or I must say the wavelengths, the lambda, okay? The wavelengths. So accordingly we use the word red, green color blindness. This is a very important thing you have to understand. So we use the word red-green color blindness for this particular disorder, what we call color blindness. Okay. So what happens in this disorder? An individual is not able to, make a distinguish or make a kind of distinction, or an individual is not able to perceive the colors. Say for example, right now I'm wearing a black shirt and it has some red strips. What happens with these individuals, or I am holding a green pen in my hand, these individuals are not able to find out the exact wavelength. They will not be able to look at that. They will not be able to recognize this as a green color. These individuals will not be able to recognize this as a red color. Although they are not blind, they will find out some hue, they will get some idea, but they will not be able to identify the red wavelength specifically. So that's why it's not called color blindness, because the individual is not color blind. Individual can perceive the color, individual can see the colors, but he or she will not be able to perceive the actual color, although they'll be able to see certain hues. Okay, so they have their kind of understanding of colors, but they don't know the exact red color, they don't know the exact green color, to be specific, because we call it red-green color blindness. This is very common. Now, my dear students, that's why if you visit some ophthalmologist, or you visit some eye specialist, or you go to some eye clinic, there the ophthalmologist is not going to use the word that the individual is suffering from color blindness. No, they'll use the word that the individual has some color vision deficiency. This is the word. Okay, this is the actual term for this disorder. Now, why isn't this individual capable of visualizing certain wavelengths? Because we know that visible spectrum is made up of wavelengths. We have blue range, we have a red range, we have a green range. So individual is not able to look at the all wavelengths. They cannot visualize all the wavelengths. Where is the defect now? That's the question. The question which arises is, where is this defect in the eye that the individual is not able to find out all the wavelengths? Where is the defect? Because we have the short wavelengths, we have the medium wavelengths, and we have the long wavelengths. Okay, say for example, the blue ones, the green ones, and the red ones. Individuals are not able, these individuals who have this particular defect, they are not able to visualize all the wavelengths. Why? Although they can visualize certain, if somebody has defect in blue range, he can visualize the other range. Now, where is the defect? That's very, very interesting. Now, my dear students, try and understand. If you look at the structure of the eye, which is going to perceive, which is going to be a sensory organ basically. So it has different regions. Okay, so we have first of all this outermost portion which is called the cornea, this particular region which is supported by the conjunctiva. So we have this cornea, and then, my dear students, we have this cavity which is filled with aqueous humor. Okay, so we have this cavity of aqueous humor, that is followed by these this muscular iris, and then that is followed by the lens. Okay, lens which is held by the ciliary body. Okay, we have ciliary ligaments over here which are going to hold this lens, and that is going to be followed by a big a vitreous humor. It's going to be a body which is composed of vitreous humor. It's a jelly kind of substance. Then that will be followed by the very important membrane which is called the retina. Okay, retina is very, very important because this is the light sensitive region of our eye. Okay, whatever is the radiations which are going to fall on the retina, because lens is going to basically do the job of focusing, then whatever the radiations are which are going to fall on the retina is going to be a kind of a sensory kind of tissue which is going to convert those light signals into the electrical signals. So I've written out that retina is the innermost light sensitive tissue. This dotted layer of green is retina basically. It's an innermost light sensitive tissue which is near the optic nerve. You can see it's near the optic nerve, and it is the tissue which is going to convert the light which is going to fall, the incident light, the photons which are going to fall on the retina. It's going to convert that energy into the neural signals, a kind of impulses, and those impulses will then be sent to the brain for visual recognition. Now what is very important is to convert these light stimuli, these photons, they'll be sensitizing, they'll be basically stimulating this retina. Now what is so special with retina? if we look at the histology, we're not going to go to the histology of the retina, but it has the different layers. But what is so important in this retina? What is so important in this retina are these cells. So retina, I'll write down over here, retina has specialized cells, which we call, number 1 are the rods and number 2 are the cones. That they are very, very important. Over here I have made only the cones. Okay, rods are not shown. So rods are basically meant for visualization of the contrast hues. Okay, they are responsible for the contrasting hues. We are basically able to look at the dim coloration because of the rods. They give us the contrast vision. Cones are responsible for the colored vision, what we call the chromaticism. So we are capable of identifying, or we are capable of distinguishing among the colors because of these specialized cells, which are the cones. So over here I have drawn 1, 2, 3 cones. What is the location of these cones? They are located in the retina. Okay, I have not given you all the cells there in the retina. Otherwise, we have different types of neurons, because this is going to be a sensory region. But what is so important in this retina are these cone cells. Rod cells are not our point of discussion right now, because we are discussing color blindness. So which are the cells which are responsible for colored vision? Are cones. Now, how many types of cones we have? We have 3 broader types of cones. We have the red cones. Since I don't have the red color, so I have used black to show you the red. So red cones, so you have to see that they help in visualizing red color. We have the green cones and the blue cones. Where is the duster? Okay, it's over here. Now, what is important as far as the red cones are concerned? So I'll write down over here. So remember that retina has rods and cones. Rods for the contrast vision, rods for the purpose of the dim vision, cones for the colored vision. Now, let us focus upon the retinal cones. Okay. Now I'll be talking about the retinal cones. Okay. Pay attention. So, let me write down now. So we have retinal cones. They contain specific pigment, which is going to be the red retinal cone pigment. So they contain specific pigment. Okay, that pigment is very much important for the perception of the red wavelength. Now, if there will be some kind of mutation to the gene which is coding for this pigment, now let me make you understand. The gene for this particular pigment, that is located on X chromosome. Try and understand. The gene for this particular pigment, which is going to be present in the red retinal cones, that gene is present on X chromosome. Now, if that gene, X chromosome, has the gene for red retinal pigment, try and understand, very easy it is. Now, if this gene is normal, the person will have the red retinal cones with the perfect pigment, and the person will be able to visualize red color. But what if this gene undergoes mutation? What if this gene undergoes mutation? In that condition, person will not be able to generate this particular pigment. So, in case of mutation to what? Mutation to the gene on X chromosome, which was coding for red retinal pigment. If that gets mutated, the individual will not be able to produce the pigment. So, no pigment. When there is no pigment, the individual will not be able to perceive red color, and therefore the individual will suffer from red color blindness. So, for example, if somebody has this mutation on X chromosome for this pigment, that means eyes of that individual will be having these red retinal cones, maybe or maybe not, but inside it there will be no pigment. So if an individual will be trying to look at this particular red color, the radiations which will be reflected from over here will reach to his or her retina, but in retina the red cone pigment is present, so the individual will not be able to find out the exact red color, although he will be able to see some different hue, maybe an orange, maybe some different hue, but not the exact red color. That condition is called the red color blindness, or we call that protanopia. That's called protanopia or red color blindness. Similarly, now let me tell you one thing. Now an individual has what? An individual, now we are talking about the green retinal cones. Earlier I was talking about red retinal cones. Now I say there are green retinal cones, right? These. Again they have the pigment. Again for that pigment you have a gene. What is the location of that gene? Again on the X chromosome. If that gene is normal, person will be able to produce the green pigment and his green cones will function and he will be able to perceive the green color. But if the same gene undergoes mutation, individual will not be able to produce the green retinal pigment. Getting my point or not? Individual will not be able to produce the green retinal pigment. Consequently, he will not be able to visualize the green color. So he will suffer from green color blindness. Okay, or we call that condition, as we call this protanopia, we call that condition Deuteranopia. What do you call that condition? Deuteranopia. We have one more condition which stands for the blue color, but that is not an X-linked condition because the gene which is going to code for the blue pigment, that's located on the autosome. So I'll tell you about that later on. That's called tritanopia. Right now we are going to talk about the red-green color blindness. So as far as this is concerned, X-linked color blindness is concerned, then we are talking about red green color blindness. Remember this point. We are not talking about blue color blindness. I'll tell you about that later on. So what is the crux over here? Retinal cones have pigments, either the red pigment, either the green pigment. Accordingly, we have the red cones and the green cones. For the pigments, we have the gene. The location of the gene is on the X chromosome. If the gene is normal, we'll be able to perceive both the colors. But if the gene undergoes mutation, then no pigment will be formed. If no pigment is formed, we will suffer from either the red color blindness or protanopia or green color blindness or deuteranopia. Okay, let me rub it off and make you understand about the genetic reasons behind it. What is happening in this position as far as the X chromosome is concerned. You must have noticed that I've written Daltonism over here. So you must be thinking about why is it called Daltonism? Why color blindness is called Daltonism? The reason is very simple. You must have heard about John Dalton. Everybody knows about John Dalton. In chemistry, you study about atomic theory and all. That was proposed by John Dalton. So my dear students, John Dalton was suffering from this particular genetic disorder. He was a colorblind individual. He had color vision deficiency, basically. Even his brother had color vision deficiency. And Dalton, so I'll write down over here, John Dalton, basically, he performed the initial studies on the heredity or the transmission of this particular disorder called color blindness. So John Dalton, that's why we call this disorder Daltonism, because it was initially investigated by John Dalton, which is a prominent name in chemistry. And he and his brother were suffering from this particular disorder. Now, my dear students, let us look at the genotypes. Okay, so we remember that this name is a misnomer. It's not color blindness, because I told you individual will be able to find out the hues but not the exact color. The correct term is to call it a color vision deficiency. Okay, so let us find out the genotypes now. So in order to find out the genotypes, we have to basically look at the genotype of a normal male, an affected male, a normal female and an affected female. Since we know that the mutation is on the X and the mutation, so we know that the mutation has happened to the X and the mutation is of a recessive type. These two things are very, very important. So if I write down something like X, this is a normal X, means the allele, the particular gene for the retinal cone pigment is normal. So I'm not writing anything else with this X. This also means that the gene is normal. So since it's XX, the individual will be definitely a female, but normal. Okay. Now I'll do something like this. I'll say there's a female who has the defect, who has this recessive mutation. So I'm giving a superscript C. C indicates that there is the mutation, there is this defect, there is this defective allele on this X chromosome. So XC represents the color blindness allele. So another X for this female is normal. This indicates that the female, you just give me the answer. Will this female be normal or she will be colorblind? She has two X. One of the X is having the mutation, one of the X is having the mutated allele. But earlier we have given this idea that this is a recessive mutation. Now this is basically a recessive form. The dominant form is over here and the dominant form is normal. So this female will be she'll be a carrier. She'll be a carrier because she has this defect, but she will be normal. She'll be normal because the condition is heterozygous. Remember I told you that this disease will appear in case of homozygous condition. So a female who has two X chromosomes and she now has mutation on one X, the recessive mutation, and her other X is normal, means the dominant allele here will mask this recessive allele and she will be herself a normal. She will not be color blind. She'll be able to perceive all the colors, although she is a carrier because she has this defect in her genome. Okay, now I'll do something like this. I'll write down X, C, X, C, means two subscripts. This X having the recessive mutation, this X having the recessive mutation. She will again be a female. Try and understand. She'll be a female, but she will be now affected with color blindness because both of the alleles are mutated and she is basically homozygous for this particular recessive mutation. She is homozygous for this recessive mutation, means both of her genes, both of her chromosomes have this recessive mutation. So have a recessive allele here because the mutation itself is recessive, but she will be able to now express the thing. She will be colorblind. She will not be able to produce the cone pigments because she is right now having a homozygous condition. Both her alleles are having this recessive type of mutation. So female will be affected only when the condition is homozygous recessive. Now what about male? Male condition is pathetic. See, male has XY. If male has one X correct, he will be a normal male, no problem at all. But what if he has this mutation? What if he has mutation on the X for the gene coding for the retinal cone pigment? What will happen? He is not having the counter X like this female. He is not having other X to mask this particular problem. So this particular condition, what we basically call this is which condition? Male has which condition? Male has the hemizygous condition. I made you understand this in my previous lecture. So in this particular situation, male is stuck and male will be affected. He will be an affected male because he has the mutation. So he will be affected male suffering from color blindness. Okay, he doesn't require to be homozygous because he is catching up the disease even in the hemizygous condition because he doesn't have the other X to mask this defective X. So in population, you will find even females affected with this particular disorder. But the proportion, the probability, the more percentage of the male will be affected because if male gets this mutation, if male receives this mutated X, he has no option. He suffers from color blindness. Same was the case with John Dalton. He and his brother both received this defective X, maybe from the mother if the mother was a carrier, or from the father if the father was colorblind. So the children, both, they were the male children, John Dalton and his brother, both of them had this defective X and they both expressed the disorder of color blindness. So you got this point now. This is X-linked and recessive. It is recessive. That's why it's not a dominant disorder because it expressed in female only in homozygous condition. It got expressed in males because male had only one X. It never gets an opportunity to express under the heterozygous condition. That's why we call it a recessive disorder. Now let us solve one numerical based on this. That's very, very important. Let us solve a question on this particular concept. There's my duster. Okay, it's here. So before I rub off this board, I just want you people to have an idea about the blue color blindness, okay? Because I have right now the cones on the board, then I'll be deleting all this stuff. So blue color blindness, when there's an individual who suffer from this, I'll write down blue color blindness, okay? Now my dear students, I'm writing with the blue color. If somebody who is having blue color blindness, he will not be able to perceive this blue color. Blue color is basically a short wavelength. Okay, it's a short wavelength. They are not able to visualize these shorter wavelengths because they have the defect in these blue cones, means they do not code the blue retinal cone pigment. That condition is called, please pay attention, that condition is called tritanopia. Okay, but that is not an X-linked defect. That is not, I just want to make it clear to you people that that is not X-linked. Okay, the gene for that cone is not the gene for this pigment, which is coding for the blue color, which is, you know, helping us in identifying the blue color. That gene is not located on X chromosome. That gene is located on your chromosome number 7. Okay, so that becomes then kind of an autosomal disorder. So tritanopia cases, you will find males and females equally affected by tritanopia. That's very important, because if there is a defect in the chromosome number 7 for this particular gene which codes for the blue retinal cone pigment, the individual will not be able to see the blue color. But chromosome number 7, the case is going to be same for males as well as for the females, because autosome is involved. That's why I told you that blue color blindness doesn't follow this particular stuff. This is wholly and solely confined to the red-green color blindness, the protanopia and the Deuteranopia case. Tritanopia is the blue color blindness, which is also called the blue color blindness or the tritanopia itself, and the gene for this particular defect, the mutation, has happened to the chromosome number 7. Now I will erase this board and I will keep a numerical ready for you people. I'll jot down the numerical and then we will solve that numerical with understanding whatever we have understood. My question is ready. It's on the board. I'll read down this question for you, but before that, let me make you understand inheritance. As far as the inheritance pattern of color blindness, what is the correct term for the color blindness is it's color vision deficiency. I've told you why we call it color vision deficiency. So inheritance pattern of this particular disorder is going to be same as hemophilia. What I've taught in hemophilia is the Criss cross inheritance, or what we call the skip generation inheritance, or what you call the zigzag inheritance. The same rule. That means if there is some father who is having color blindness, father gives this trait to the daughter, and daughter then transfers this trait to the son. Okay, so from father it goes to the grandson. Okay, this type of transmission is called the skip generation transmission. We have discussed this in detail in our hemophilia. This is the same point. If father has the defective X, that will go to the daughter. Daughter will become the carrier, and if she gives the defective X to the son, son automatically attains or acquires this particular trait. So the transmission takes place from father to daughter to son, where daughter is the carrier but son gets affected. Now you will be able to understand this again. This particular transmission you will be able to understand. I have selected this numerical to give you understanding of the same fact also. So numerical says if a colorblind male, so we have a color blind male. Go to your genotypes. If we work for the genotype of a colorblind male, how it will be? It will be X, Y, but X with the mutation. Then it's the colorblind male. Okay, colorblind male. If a colorblind male marries a normal female he is saying, but in the bracket there is written heterozygous. Pay attention. So female is heterozygous. That's very, very important. Female is heterozygous, normal female. That is the problem. means she is not like XX, she is X raised to power C. That means she is heterozygous. She has the recessive mutation, but she has the correct dominant allele. So this is the genotype for the heterozygous normal female. She will be herself normal, but she will not express the trait of color blindness, but she basically is the carrier. She is having the heterozygous condition. She has the mutation on one X chromosome. Whereas the male, the father, the husband of this particular woman is going to be a colorblind. So we have shown that this is the representation, genotypic representation of a colorblind male. So first and foremost, before solving the questions, you must be able to find out the genotypes of the couple. If a colorblind male marries a normal female, which is heterozygous, so we have worked, our frame is ready. The question is, what is the probability that the child is colorblind? What is the probability that the child is male and colorblind? If the child is female, what is the probability that she is normal? If the couple has 2 children, what is the probability that both of them are normal? So this will help us to understand the inheritance pattern. Also, if anybody of you has this problem in understanding, you'll be able to work on this numerical and find out the answer. This is going to be very, really interesting. So let us first of all work with the each and every probability. So this male will produce the gametes, female will produce the gametes. So we have the sperms and the ova. Let us draw out a Punnett square quickly. So this is the Punnett square over here. Very, very important. I want my Punnett square to be, absolutely. Okay. So let me draw the Punnett square. I have to be very careful because the lines become tilted. Okay. This is it. Oh, Alhamdulillah. It has now become suitable. Okay. I always draw improper lines. Okay. Okay. This is quite presentable, I think. Yeah, it is. It will serve the purpose. Okay. Let me make it proper. Okay. So here it is. So on my Punnett square, I'll put down the female gamete this way and the male gametes this way. So how many types of sperms the colorblind male will be able to produce? 2 types of sperms. One sperm having this defective X, another sperm carrying the Y. Okay. How many types of ova carrying the correct, not the correct, it's the defective X, another ova carrying the correct form of the X, no mutation over here. So we have the 2 types of ova. So we have these 2 types of ova over here. And we have these 2 types of sperms over here. Now let me find out the probability. Let me find out the fertilization coinsequence. So this is the representation of an ova and ova. So this is the sperm. This is the sperm. Now we have to talk about the fertilization event. If this type of sperm fertilizes this ova, the product is over here. So we'll have X with defect, X with the defect. Okay. What type of children it will be? It will be a female, that too colorblind. Clear? If this sperm fertilizes this type of ova, product will be over here. So we'll have X normal and this X defective. So XC, X normal. Again, she will be a female, clear from the genotype, and she will be a carrier. That means she will be heterozygous. But she will not be a colorblind. She will be normal. So we can write down she is normal. But we have to remember the fact that she is a carrier. She is heterozygous like her mother. Okay. Now as far as males are concerned, let me write down the males, the other color. So as far as males are concerned, that will confuse you people. Okay. So XC, if this sperm fertilizes this, then we have X with subscript C and Y. This and this. The product is over here. XY. It's going to be a male, but no opportunity to be safe. He'll be color blind. If this and this fertilize, it will be X, Y. This be again a male, but he'll be absolutely normal. So we got all the possibilities. 50% chance for the female, 50% chance for the male. There will be one female probability. It's the probability distribution basically. There will be one female out of the 4 who will be colorblind. one female out of the 4 who will be, out of the 4 children who will be carrier, one male out of the 4 children who will be affected male, one male out of the 4 children who will be a normal male. Now, putting this into consideration, we'll be able to solve these. These are very simple now. Look, try and understand. What is the probability that the child is colorblind? What is the probability that the child is colorblind? Can you find out where is the case when the child is born a colorblind? We have 2 cases for colorblind. We can have, I'll use this pen, we can have this female colorblind and we can have this male colorblind. That means out of 4 children, we have 2 children out of 4. The probability says we have 2 children out of 4 children which could be born colorblind. That implies how much? That implies 0.5. That is equivalent to 50%. That means there are 50% chances, there is 50% probability that the child is born colorblind. You're getting my point or not? So this is for part A. I said there are 2 children out of this Punnett square. This is normal, this is normal. 2 children out of how many children? 4 are colorblind. So this gives out the value of 0.5, and that's what is the probability that the child is male and colorblind? Very simple. What is the probability that the child is male as well as colorblind? So for that case, I'll find just this male fitting this situation. Out of the 4 children, there's only just this male who is, first of all, male as well as color blind. So out of 4 children for part B, I'll say there is just 1 male who is color blind. So I'll say 1 out of 4 is the probability. That means 0, no, it's not 0, this will be 0.25. 0.25. This is equivalent to 25%. So there is 25% probability that the child is male and colorblind. If we put all the children into consideration, there is the probability, 25% probability for 1 child to be male as well as colorblind. Part C says if the child is female, what is the probability that she is normal? So we have to categorically omit the males. We are not talking about males because the question says if the child is female. So we have to focus only the females. What is the probability that she is normal? So we have probability of females is, we have 2 females. Out of 2, how many of them are normal? Only 1 is normal. So we'll say 1 female out of how many females? 2. There's a probability that 1 among the 2 will be normal. So that means if the children is female, if the child is female, what is the probability that she is normal? 1 among the 2. Out of the 2 females, 1 is normal. So 1 among the 2 will be the normal female. This is the basically colorblind female. We're talking about this female, okay? So 1 among the 2, which is equal to 0.5. That is equivalent to 50%. That means if the child is female, then we have 2 females. Of the 2 females, there is 50% probability that the female will be normal. As simple as that. Part D says if the couple has 2 children, what is the probability that the child, both of them are normal? So here we have to work for child 1 and child 2. Let us first of all work for child 1. What is the probability that child 1 will be normal? As far as part 1 is concerned, what is the probability for the child to be defective or colorblind? It's 50%. So accordingly, we can use the common senses. If there are 50% chances for the child to be colorblind, there will be 50% probability for the child to be normal. As simple. That means 0.5. Okay. Applying the same logic for child 2. Again, we have 50% probability for child 2 to be normal. Getting my point or not? So that means again 0.5. So 0.5 or 50% probability for 1 child to be normal, 50% probability for 1 child to be normal. This is child 1, this is child 2. This stands for the question if the couple has only 2 children, what is their probability to be normal? Now, both of the events are independent of each other. If this child is born, there's 50% probability that the child will be normal. If this child is born, there's 50% probability for the child to be normal. Now, we have to find out the overall probability for the 2 children to be normal. So the final probability, we can say that the actual probability, we can call it actual probability will be equivalent to the multiplication of the 2. That is 0.5 into 0.5. That is equivalent to 0.25, which is equivalent to 25%. So there is, if the couple has 2 children, there is 25% probability that both of those children will be normal. So in this way, you got an idea about how to solve these pedigree based questions, or how to solve these numericals on, the same type of numerical could be asked for hemophilia also. Now, as far as, my dear students, where is my duster? As far as this skip generation, so sorry, this is not skip generation, this is skip, okay. This is skip generation. As far as skip generation is concerned, I have given you the detailed explanation for skip generation in hemophilia. Over here, let me quickly give you a kind of idea once again. So, skip generation means what? The mother is normal. So we have a normal, I'll say normal female, pay attention. A normal female, and I'll say a color-blind male. What are the genotypes? I'm making you understand this particular thing. Although you got to know about the depth over here, let you have an idea about skip generation, whereby I'll make you understand that the father gives to the daughter and daughter gives this trait to the son. You'll be able to understand this is skip generation, okay? So we have a normal mother and an affected father. So normal mother will show like XX, and affected father will show by XCY as over here, okay? So they marry, she'll be able to produce gamete, normal gamete over carrying X. Father will produce 2 types of sperms, one carrying defective X, another carrying normal Y. What is the probability if this and this will fuse? We'll have X, we'll have the girl, and if this and this will fuse, we'll have the boy. So in this marriage, if a marriage happens between the normal mother and an affected father, who becomes affected now? Father, this was the father, he was affected, he gave this trait to the daughter. But daughter is what? Is she carrier or affected? She is a carrier. Now, if this carrier daughter, this is what skip generation means. Trait got transferred, but it didn't express, okay? So, if this daughter marries now a normal male. So, suppose she marries a normal male, okay? This is a male which is normal and she married to her. Now, look, where will the trait go? Father will produce X and Y, okay, sperms. Mother will produce 2 types of X. X with mutation, X normal. If this ova will fertilize this Y, we will have X, Y. And over here, the son will be affected. Suppose if this ova fertilizes this X, again we will have XCX. We will have the female, but she will not be affected, she will be the carrier. Who will be affected? Son will develop the color blindness. Can you follow the track? Can you follow the track? It was the grandfather, okay? This was the father, this is his daughter, and this is the son. So this is whose grandson? This is this man's grandson. So one generation got skipped. So such a type of transmission where you have a father transferring his trait to the daughter and daughter giving this trait to her son, you call this transmission crisscross, skip generation, or zigzag inheritance. I have made you solve the numerical also. This will help you in your examination. This was all for today. Inshallah, in next video lecture, I'll come back with some other topic. Don't forget to subscribe to my channel and share this video with your friends and colleagues so that everybody will understand and will expand our family of AmreenaMehrajTutorials. Thank you so much. Take care. Fi amanillah.